This executive scientific report provides an overview of the repurposing of covalent inhibitors as warheads for product design. It highlights key trends, leading companies, and future directions in this rapidly evolving field. 1. Introduction. Proteolysis targeting chimeras PROTACs offer a groundbreaking approach to selectively degrade disease-causing proteins using the 𝗖𝗢𝗩𝗪𝗓𝗓 system. Covalent inhibitors, known for their high affinity and irreversible binding to target proteins, are increasingly repurposed as warheads in product molecules. This strategy improves target engagement and degradation efficiency, offering a promising avenue to expand the therapeutic reach of PROTACs, particularly for traditionally undruggable targets. 2. Top covalent inhibitors repurposed as product warheads. Several FDA and EMA-approved covalent inhibitors are being leveraged as warheads in product designs, with clinical stage compounds emerging from this strategy. A. Ibrutinib 𝗥𝗘𝗣𝗨𝗥𝗑𝗘𝗧, acrylamide-based 𝗥𝗡𝗛𝗅 covalent warhead targeting cysteine residues. Target: Bruton's tyrosine kinase BTK. Status: FDA approved for various hematological malignancies. Sales approximately for $0.2 billion in 2023. Product development: several BTK product candidates in clinical and preclinical phases utilize ibrutinib-derived warheads for enhanced degradation of BTK in resistant cancers. B. Osimertinib 𝗤𝗏𝗏𝗏, chemistry: electrophilic acrylamide group covalently modifying EGFR mutant cysteines. Target: EGFR T790M mutation in an SCLC. Status: FDA and EMA approved. Sales approximately $6 billion in 2023. Product development: osimertinib-based PROTACs are advancing in early clinical studies, aimed at overcoming resistance mechanisms by targeted protein degradation rather than inhibition alone. C. Afatinib gilteritinib, chemistry: covalent quinazoline scaffold with acrylamide warhead. Target: EGFR family kinases. Status: FDA and EMA approved for NSCLC. Sales approximately $1.1 billion in 2023. Product development: afatinib-derived warheads are being integrated into product platforms targeting EGFR-driven cancers, with preclinical data supporting effective degradation. 3. INDs leveraging covalent warheads for PROTACs. Emerging IND stage product candidates use covalent inhibitors as warheads to improve selectivity and potency. BTK PROTACs. Various developers, including Arvinas and Nurix, warheads derived from ibrutinib and analogs, using 𝗥𝗘𝗪𝗪𝗓𝗏𝗪 targeting cysteine residues. Phase 1 INDs ongoing for resistant B-cell malignancies. EGFR targeting PROTACs. AstraZeneca, C for Therapeutics, warheads derived from osimertinib and afatinib, using covalent acrylamide groups. Early clinical INDs targeting NSCLC and other EGFR-driven tumors. Novel PROTACs targeting other kinases, utilizing covalent warheads derived from approved inhibitors, for example, KRASG1 to C covalent inhibitors, with submissions expected within 1 to 2 years. 4. Chemistry types and mechanistic advantage. Escalent warheads used in product design typically involve 𝗥𝗘𝗪𝗪𝗓𝗏 acrylamides that react with cysteine residues on target proteins, forming irreversible bonds, electrophilic groups that enable durable target engagement and increased degradation efficiency, reversible covalent warheads that offer a balance between irreversible binding and controlled off-target effects. The covalent nature enhances product residence time and selectivity, crucial for potent target degradation and reduced off-target toxicity. 5. Leading players in covalent warhead-based product design and development. Key companies pioneering covalent warhead PROTACs include Arvinas, Inc., a leader in clinical stage PROTACs, including BTK-targeted PROTACs using covalent warheads. The company offers integrated drug discovery platforms combining medicinal chemistry and proteomics. Nurix Therapeutics, focused on E3 ligase modulation and covalent product warheads for hematological cancers. Nurix maintains strong partnerships with major pharmaceutical companies. AstraZeneca, developing covalent warhead PROTACs targeting EGFR and other kinases. Leveraging expertise in covalent kinase inhibitors and product technology, 𝗖𝗢𝗩𝗕𝗚𝗓 Therapeutics, specialized in product technology integrating covalent chemistry for kinase and transcription factor degradation. These companies collaborate extensively with contract research organizations cross-specializing in covalent chemistry and product linker design, including Biotech, 𝗥𝗘𝗣𝗥𝗡𝗔𝗡, and Pharma. 6. Market analysis and economic projection. Sales of covalent inhibitors repurposed as product warheads. 2022-2024. Ibrutinib-based therapies generated approximately $4.02 billion annually. Osimertinib-based therapies generated approximately $5.88 to $6 billion annually. Afatinib-based therapies generated approximately $1 to $1.1 billion annually. Given the increasing number of product candidates utilizing these covalent warheads, the market value is expected to grow substantially. Projected product market value 2025-2027. Ibrutinib-derived PROTACs are projected to reach $2.50 to $4.5 billion, driven by improved outcomes in resistant hematologic malignancies. 7. Economic valuation and sensitivity analysis of end product assets assuming successful clinical development and approval within 3 years. Estimated economic valuations, net present value, NPV for leading end PROTACs with covalent warheads are as follows. The TK product Ibrutinib warhead, estimated peak sales between $2 billion and $4.5 billion. NPV ranges between $8 billion and $14 billion. Sensitivity analysis with plus or minus 20% variation results in a range from $6.4 billion to $16.8 billion. EGFR product Osimertinib warhead, estimated peak sales between $3 billion and $5.5 billion. NPV ranges between $10 billion and $16 billion. Sensitivity analysis with plus or minus 20% variation results in a range from $8 billion to $19.2 billion. EGFR product Afatinib warhead, estimated peak sales between $1 billion and $2 billion. NPV ranges between $4 billion and $7 billion. 8. Conclusion: repurposing covalent inhibitors as warheads in product design represents a transformative strategy to enhance targeted protein degradation. The clinical and commercial success of FDA and EMA-approved covalent inhibitors such as Ibrutinib, Osimertinib, and Afatinib validate the chemical scaffolds and target engagement mechanisms critical for effective PROTACs. With robust pipelines and growing investment, the next 3 to 5 years are poised to deliver significant breakthroughs in targeted degradation therapies driven by leading biotech and pharmaceutical innovators.